NCERT single sentence questions, excretory products and their elimination. Animals accumulate ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium, potassium, chloride, phosphate, sulfate, either by metabolic activities. In what way? Either in the form of metabolic activities or by other means like excess ingestion, either in the form of excess ingestion or a metabolic activities. They release sodium, potassium, chloride, phosphate, sulfate, etc. Major forms of the nitrogenous wastes excreted by the animals are ammonia, urea, and uric acid. Then most toxic nitrogenous waste among these, ammonia. Least toxic nitrogenous waste among these, ammonia, urea, and uric acid. Least toxic waste will be uric acid. Toxic waste material require large amount of water for its elimination. Ammonia, it's a highly toxic, that's why it require more water. Toxic waste material which can be removed with a minimum loss of water, uric acid. Some amount of dash may be retained in the kidney matrix of some animals to maintain desired osmolarity. Some amount of urea we maintain. The process of excreting ammonia is called ammonotelism. The process of excreting urea is called ureotelism. Next, the process of excreting uric acid is called uricotelism. Then identify the major excretory material of given animals. Many bony fishes excrete ammonia, and mammals will find urea. Reptiles are excreting uric acid. Identify the major excretory material of given animal. Many terrestrial amphibians are going to excrete urea, and aquatic amphibians are excreting ammonia. Then birds are excreting uric acid. Excretory material of birds is uric acid. Land snails, excretory material of land snails. Land snail excrete urea. Then marine fishes excrete urea. Aquatic insects excrete ammonia. Insects excrete uric acid. Terrestrial insects excrete uric acid. Example for ammonotelic animal. As we said that above, that many bony fishes, aquatic amphibians, and aquatic insects are excreting ammonia. We call them as ammonotelic animal. Ureotelic animal. Examples for ureotelic animal: mammals and many terrestrial amphibians. Then marine fishes are excreting urea. We call them as ureotelic animals. Example for uricotelic animals. Reptiles are excreting, and birds, land snails, and insects. Excretory material which is readily soluble and generally excreted by the diffusion across body surface. Ammonia. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous wastes like urea and uric acid, or lesser toxic material. Excretory waste which eliminated through the gill surfaces in fish is ammonium ions. In ureotelic animals, dash or produced by metabolism is converted urea into the, that in ureotelic animal, ammonia produced by metabolism is converted urea in the liver, and released into the blood, which is filtered and excreted out by the kidneys. Kidneys do not play any significant role in the removal of this excretory waste material. Kidneys do not play any significant role in the removal of this excretory material, ammonia. Uricotelic animals excrete nitrogenous wastes as uric acid in the form of pellet or paste, and that makes a minimum loss of water. Uricotelic animals excrete nitrogenous wastes as uric acid in the form of pellet or paste with minimum loss of water. In most of the invertebrates, excretory structures are simple and tubular forms, whereas vertebrates are having complex tubular organs we call kidneys. Malpighian tubules help in the removal of Malpighian tubules help in the removal of nitrogenous wastes like uric acid, and they are helpful for osmoregulation process. Excretory structures of platyhelminthes are protonephridia, we can call, or flame cells. Protonephridia or flame cells. Excretory structures of rotifers and some annelids. Protonephridia. Excretory structures of cephalocardate like Amphioxus. Protonephridia, we call them as solenosides. Protonephridia are primarily concerned with ionic and fluid volume regulation. That is called osmoregulation. Dash are the tubular excretory structures of earthworms and other annelids. Dash are the tubular excretory structures of earthworms and other annelids. Nephridia. Structures which help to remove nitrogenous wastes and maintain fluid and ionic balance in earthworms and some annelids. Nephridia. Excretory structures of most of the insects, including cockroaches. Malpighian tubules. Function of excretory organ in most of the insects, including cockroaches. Removal of nitrogenous wastes along with osmoregulation process. Removal of nitrogenous wastes and osmoregulation. Dash perform the excretory functions in crustaceans like prawns. Antennal glands or green glands, we can say. Inner to the hilum is a broad funnel-shaped space called renal pelvis, and which projects with projections called calyces. Excretory system of human consists a pair of kidneys. That human being excretory system contain a pair of kidneys and a pair of ureters and a urinary bladder, then urethra. Shape and color of the kidney. Shape is bean shape, and color reddish brown. Kidneys are situated between the levels of last thoracic and third lumbar vertebrae. Kidneys are situated between the levels of last thoracic and third lumbar vertebrae, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeter in length and 5 to 7 centimeter in width, then 2 to 3 centimeter in thickness, 120 to 170 grams in weight. The point at which the renal artery and nerves enter and renal vein and ureter leave is called hilum. Towards the center of the inner concave surface of the kidney is a notch called hilum. Outer layer of the kidney is covered by a tough capsule which is made up of irregular, dense, fibrous connective tissue. Two zones of the kidney inside are the two zones are outer zone called cortex and inner zone said to be medulla. Outer surface of the kidney is convex. Outer portion of the kidney is called cortex. Inner portion of the kidney is called medulla. Functional units of kidney are nephrons. Multiple cone-shaped masses of tissues of medulla are called medullary pyramids. Medullary pyramids. Multiple cone-shaped masses are called medullary pyramids. Single form of calyces is calyx. Number of nephron in each kidney, 1 million. Medullary parts projecting into the cortex are called columns of Bertini. Columns of Bertini. The cortex extends in between the medullary pyramids as renal columns called. The cortex extends in between the medullary pyramids as renal column is called columns of Bertini. Columns of medullary parts projecting into the calyces. We have a small mistake here. These are renal pyramids. In between the renal pyramids, cortical extension we call columns of Bertini. Medullary part projecting into the calyces. Cortical portion projecting into the medullary pyramids. Columns of Bertini. Medullary part projecting into the calyces are called renal pyramids. Complex tubular structures of kidney are called nephrons. Each nephron has two parts. What are they? Glomerulus and renal tubule. Tuft of capillaries formed by the afferent arteriole is called glomerulus. Double-walled cup-like structure of renal tubule is Bowman's capsule. Blood from the glomerulus is carried away by an efferent arteriole. Renal tubule begins with Bowman's capsule. Branches of this artery form the glomerulus. Branches of renal artery is going to form the glomerulus. Tuft of capillaries are enclosed by dash of renal tubule. Tuft of capillaries are enclosed by Bowman's capsule of renal tubule. Bowman's capsule continues further to form a highly coiled network called proximal convoluted tubule. Hairpin shaped structure of renal tubule is Henle's loop. Hairpin shaped structure, Henle's loop. Glomerulus along with Bowman's capsule is called Malpighian body. Proximal convoluted tubule further opens into descending limb of Henle's loop. The ascending limb of Henle's loop continues as another highly coiled tubular region called distal convoluted tubule. Descending limb of Henle's loop continues as ascending limb. The DCTs of many nephron open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the collecting duct. Then renal pelvis. The DCTs of many nephron open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. Hairpin shaped Henle's loop has a descending limb. Hairpin shaped Henle's loop has a descending limb and ascending limb. Part of Malpighian body which belongs to circulatory system. Part of Malpighian body which belongs to circulatory system, glomerulus. Part of Malpighian body which belongs to renal tubule, Bowman's capsule. Malpighian body is also called as renal corpuscle. Renal corpuscle. Malpighian corpuscle, PCT and DCT of the nephron are situated in the cortical region. Of the kidney, whereas the loop of Henle dips into the medulla. In majority of nephron, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. Vasa recta is absent or highly reduced in cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called peritubular capillaries. The minute vessel of this peritubular network runs parallel to the Henle's loop, forming a U-shaped blood vessel called vasa recta. Recognize the type of nephron based on the given character as Malpighian corpuscle, PCT and DCT in the cortical region. So, A, cortical nephrons. In case of cortical nephron or juxtamedullary nephron, where do you find this? Malpighian corpuscle, BCT and DCT in the cortical region. So, in both cortical nephron and medullary nephron, Malpighian corpuscle, PCT and DCT are present in the cortical region only. Henle's loop is too short, cortical nephron. Henle's loop is too long, medullary nephron, juxtamedullary nephron. Majority in number, cortical nephron. Less in number, juxtamedullary nephron. Efficient water absorption is possible with counterflow mechanism, juxtamedullary nephron. Urine formation involves three main processes. The three main processes we can say glomerular filtration, and reabsorption process, then secretion, tubular secretion or augmentation we can say. The first step in urine formation is filtration of blood. Then which is carried out by the glomerulus is called glomerular filtration. Then blood pumped out by each ventricle of the heart in a minute can be termed as cardiac output. Amount of blood filtered by the kidney per minute is 1100 to 1200 ml. The cardiac output of human approximately is 5040 ml. In case 70 stroke volume, 72 heartbeats per minute, 5040 ml. On an average, that how much blood on average? 1100 to 1200 ml of blood reaching the kidney. On an average, 1100 to 1200 ml blood filtered by the kidneys per minute, which constitute roughly one-fifth of the blood pumped out by each ventricle of the heart in a minute. The dash causes filtration of blood through three layers. Glomerular capillary blood pressure. The dash of glomerular blood vessels, the endothelium of Bowman's capsule and epithelial membrane, the endothelium of glomerular blood vessel, epithelium of simple squamous epithelium of Bowman's capsule, and a basement membrane. And a basement membrane between these two layers. We call it as filtration membrane. The epithelial cells of Bowman's capsule are called podocytes, arranged in intricate manner. Okay, arranged in intricate manner, so has to leave some minute spaces called filtration slits. Filtration slits or slit pores. Filtration slits are slit pores. Blood is filtered so finely through these three membranes that almost all the constituents of the plasma except the proteins, pass on to the lumen, of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. Process we call ultrafiltration. Pressure created due to the diameter difference between the afferent and efferent arteriole is 60 mm of Hg. 60 mm of Hg. Then blood colloidal opposing osmotic pressure is 32 millimeter of mercury. Bowman's capsular hydrostatic opposing osmotic pressure is 18 millimeter of mercury. Total opposing pressure 50 mm of Hg. Total opposing pressure. Net filtration pressure is 10 mm of Hg, which is helpful for filtration. Blood filtrated through the fine slit pores of Bowman's capsule and fenestrations of blood capillary due to net filtration pressure. This process of filtration is called as ultrafiltration. This process of filtration called ultrafiltration. Amount of blood that enters into the kidney from the total cardiac output is, as we said, it has 1/5 volume, 1/5, 1100 to 1200 ml. The amount of filtrate formed by both kidneys per minute is called glomerular filtration rate. Glomerular filtration rate. Amount of urine excreted by normal human in a 24-hour period, that 1 to 1.5 liter. Glomerular filtration rate per minute in normal individual, 125 ml per minute. Okay, glomerular filtration rate per day in normal individual. Per day it is going to be 180 liters. Amount of blood enters into the kidneys per day. Amount of blood enter into the kidney per day, it is going to be 1584 liters to 1728 liters. As we calculated in the form of 125 ml per minute, so that means per day how much? Like percentage of urine excreted out from the GFR. Percentage of urine excreted from the GFR is 1% only. Percentage of reabsorption possible from the total GFR, 99% we absorb. The kidneys have built-in mechanism for the regulation of glomerular filtration. Regulation of glomerular filtration rate. Then one such efficient mechanism is called juxtaglomerular apparatus. Juxtaglomerular apparatus. JGA, juxtaglomerular apparatus, is a special sensitive region formed by the cellular modifications in the distal convoluted tubule, and the afferent arteriole. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow. And thereby the GFR back to the normal. Macula densa together with JG cells form juxtaglomerular apparatus. Juxtaglomerular cells of JG apparatus are modified smooth muscles of afferent arteriole in JG apparatus. Modified smooth muscles of afferent arteriole, juxtaglomerular cells. A group of modified epithelial cells of the DCTR which are crowded where they connected to afferent arteriole is called macula densa. Modified smooth muscle cells of the afferent renal arteriole where they connected to the DCT are juxtaglomerular cells. Juxtaglomerular cells. A comparison of the volume of the filtrate formed per day 180 liters per day with that of the urine released 1.5 liters every day suggests that nearly 99% we absorb. 99% of the filtrate has to be reabsorbed by the renal fluid. This process is called as reabsorption. Reabsorption, we say it as. The tubular epithelial cells in different segments of the nephron perform this either by active process or passive process. Substances like glucose, amino acids, sodium ions, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous wastes are absorbed passively. Reabsorption of water also occurs. Reabsorption of water also occurs passively in the initial segments of the nephron. During urine formation, the tubular cell secretes substances like secretion of the substances. It may be by H plus ions or we can say potassium ions and ammonia. H plus, potassium and ammonia. Augmentation, we can say tubular secretion. We can call it as augmentation or tubular secretion. Tubular secretion is also an important step in urine formation. It helps the maintenance of ionic and acid base balance of the body fluids. That is, we call it as dash is also an important process. We can say a tubular secretion or augmentation process. We need to say it as augmentation process. If substances of filtrate moved out from the renal tubule into the interstitial fluid of the kidney, such process is called reabsorption. If the substances are moved into the filtrate to maintain ionic or acid balance, such process is said to be, as we said, about tubular secretion, absorption and secretion. Part of nephron which is lined by simple cuboidal brush border epithelium and advantage. Part is going to be proximal convoluted tubule. We can say proximal convoluted tubule, and it absorbs. It increases. It enhances the absorption of. It increases area of water for area for reabsorption. Area for reabsorption. Identify the part of nephron. Reabsorption is minimum in its. Reabsorption is minimum in its ascending limb of Henle's loop. Reabsorption is minimum. Identify the part of nephron. Nearly essential nutrients, 70 to 80% of electrolyte and water reabsorbed in proximal convoluted tubule. Then it also helps maintain the pH and ionic balance of the body fluid by selective secretion of hydrogen ion, ammonia and potassium ion into the filtrate and by absorption of bicarbonate from it. It will be proximal convoluted tubule itself again. Identify the part of nephron. This region plays a significant role in the maintenance of high osmolarity of the medullary interstitial fluid. High osmolarity of the medullary interstitial fluid. That is ascending limb of Henle's loop. Ascending limb of Henle's loop. Concentration gradient increases due to the absorption of water into the medullary fluid, that is descending limb of Henle's loop. Conditional reabsorption of sodium and water takes place in this segment, that is in conditional reabsorption occur in distal convoluted tubule. Then concentrated filtrate pass upward, it gets diluted due to the passage of electrolytes to the medullary fluid, that is happening in ascending limb of Henle's loop. Ascending limb of Henle's loop. Identify the part of nephron. It is also capable of reabsorption of bicarbonate and selective secretion of hydrogen and potassium ion and ammonia to maintain pH and sodium potassium balance in blood. That is happening at distal convoluted tubule. Identify the part of nephron. This long duct also play a role in the maintenance of pH and ionic balance of the blood by selective secretion of H plus and potassium ion. That is collecting duct. Collecting duct. Identify the part of nephron. Concentration gradient decreases due to the absorption of electrolytes into the medullary fluid. That is ascending limb of Henle's loop. Identify the part of nephron. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Inner parts of the medulla. Collecting duct. Identify the part of nephron. It is permeable to water but almost impermeable to electrolyte. These concentrate the filtrate as it moves down descending limb of loop of Henle. Identify the part of nephron. It is impermeable to water but allows transport of electrolytes actively or passively. Ascending limb of loop of Henle. Ascending limb of loop of Henle. Next, identify the parts of nephron. Large amount of water could be reabsorbed from this region to produce concentrated urine. The segment allows passage of small amounts of urea into the medullary interstitium to keep up the osmolarity. Collecting duct. Collecting duct. Identify the part of nephron. Secretion of H plus and ammonia. Secretion of H plus and ammonia is given in NCERT, PCT. Absorption of water only in NCERT, it is given descending limb of Henle's loop. Descending limb of Henle's loop. Identify the part of nephron. Absorption of sodium chloride and secretion of urea. Thin segment of ascending limb of Henle. Identify the part of nephron. Absorption of sodium chloride. Absorption of sodium chloride. Thick segment of sodium chloride. Thick segment of ascending limb of Henle's loop. Ascending limb of Henle's loop. Absorption of sodium chloride, water, bicarbonate, and secretion of potassium and H plus ion. That is distal convoluted tubule. Absorption of water and urea. Absorption of water and urea. That collecting duct. Mammals have the ability to produce a concentrated urine. The concentrated urine. The dash play a significant role in this. The dash and dash play significant role in these. Henle's loop and vasa recta. Henle's loop and vasa recta play a significant role in this area. The flow of the filtrate in the two limbs of Henle's loop is in opposite direction and thus forms a counter current. The flow of blood through the two limbs of vasa recta is also in counter current pattern. Counter current pattern. The proximity between the Henle's loop and vasa recta as well as the counter current in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is 300 milliosmol per liter in the inner medulla. This gradient is mainly caused by sodium chloride and urea. That's it. Sodium chloride is transported by the ascending limb of Henle's loop, which is exchanged with the descending limb of vasa recta. Sodium chloride is returned into the interstitium by the ascending portion of vasa recta. Small amounts of urea enter the thin segment of the ascending limb of Henle's loop, which is transported back to the interstitium by the collecting tubule. Okay, the above described transport of substance is facilitated by the special arrangement of Henle's loop and vasa recta is called counter current mechanism. Counter current mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient by counter current mechanism helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate in collecting tubule. Okay, collecting tubule is given here actually. The filtrate, collecting tubule, and then the filtrate, what do you call the filtrate as? Urine. Human kidneys can produce urine nearly 4 times concentrated than the initial filtrate form. Mammals have ability to produce concentrated urine. The structure of renal tubule and peritubular net helpful for this particular process. Henle's loop and vasa recta are helpful by making counterflow mechanism. Based on the flow of renal fluid, Henle's loop is classified as descending limb and ascending limb. Fluid goes down and fluid goes up. Part of Henle's loop where renal fluid is from outer medulla to inner medulla. Descending limb. In the descending limb, it is happening. Flow of renal fluid from outer medulla to inner medulla. Part of Henle's loop where the flow of renal fluid from inner medulla to outer medulla. Ascending limb. Based on the flow of blood, vasa recta is classified as same ascending limb and descending limb. Part of vasa recta where the flow of blood is from outer medulla to inner medulla, descending limb. Part of vasa recta where the flow of blood is inner medulla to outer medulla, ascending limb. Flow of renal fluid in the descending limb and ascending limb of Henle's loop is opposite to the flow of blood in the ascending limb and descending limb of vasa recta, respectively. Such a special kind of flow of fluids is called counterflow. Flow of renal fluid in the descending limb of Henle's loop is opposite to the flow of blood in ascending limb of vasa recta. Flow of renal fluid in the ascending limb of Henle's loop is opposite to the flow of blood in descending limb of vasa recta. The flow of renal filtrate in the two limbs of Henle's loop is in opposite direction. The flow of renal filtrate in the two limbs of Loop is also in opposite direction. Blood is also in opposite direction. The two limbs of vasa recta. Reason for decrease in concentration gradient in ascending limb of Henle's loop from the inner medulla to outer medulla due to absorption of sodium chloride into medullary fluid. From there, vasa recta. Reason for the increase in the concentration gradient in descending limb of vasa recta from outer medulla to inner medulla. It absorbs sodium chloride and urea from descending limb of vas and collecting duct, descending limb of Henle's loop and collecting duct via medullary fluid. Okay, descending limb of Henle's loop and collecting duct via medullary fluid. Reason for the decrease in the concentration gradient of ascending limb of vasa recta from inner medulla to outer medulla. From inner medulla to outer medulla. It absorbs water from descending limb. It absorbs water from descending limb of the Henle's loop via medullary fluid. Reason for increase in the concentration gradient of descending limb of Henle's loop from outer medulla to inner medulla. Outer medulla to inner medulla. Water is absorbed into the ascending limb of vasa recta. Ascending limb of vasa recta via medullary fluid. To absorb more water, blood of vasa recta need to maintain hypertonic condition. How did they maintain? Ascending limb of vasa recta absorb sodium chloride and urea. They absorb sodium chloride and urea. It facilitates the blood to become hypertonic. In ascending limb of vasa recta to absorb more water. Ascending limb of vasa recta to absorb more water. That's sodium chloride absorbed by descending limb of vasa recta is returned back to the interstitium from ascending portion of vasa recta. In return, they give ascending portion of vasa recta. Transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called counter current mechanism. Special mechanism is called counter current mechanism. Hormone stored in neurohypophysis and produced by hypothalamic neurons. Antidiuretic hormone, ADH, because hormone released from the mineralocorticoids of adrenal cortex, aldosterone. Enzyme produced from the juxtaglomerular cell, renin. Juxtaglomerular cells of the juxtaglomerular apparatus secrete renin. Okay, then antidiuretic hormone is also called, okay, wait here, a protein produced in the liver which is helpful to raise GFR and blood pressure. Angiotensinogen. Antidiuretic hormone is also called vasopressin. Angiotensin converting enzyme is produced from lungs. Atrial natriuretic peptide is produced from. ANF is from the right atrium of the heart. Functioning of the kidney is efficiently monitored and regulated by hormonal feedback mechanism, we can say hormonal feedback mechanisms involving the hypothalamus and juxtaglomerular apparatus, and to certain extent the heart by releasing ANF, atrial natriuretic factor. An increase in volume of the body fluid can switch off the osmoreceptor and suppress the release of ADH. Such mechanism is called negative feedback, we can say. It is called negative feedback mechanism. Decrease in the volume of the body fluid can activate the osmoreceptors, which in turn they facilitate the release of ADH. Such mechanism is called positive feedback control. Positive feedback control mechanism. Then this enzyme catalyzes the conversion of angiotensinogen into angiotensin 1 renin from juxtaglomerular cell. Enzyme which convert the angiotensin 1 into angiotensin 2. Angiotensin converting enzyme which is released from lungs. Angiotensin converting enzyme released from lungs. Asthma receptors in the body are activated by changes in blood volume, body fluid volume, and ionic concentration. An excessive loss of fluid from the body can activate the osmoreceptor, which stimulate the hypothalamus to release a hormone called antidiuretic hormone or vasopressin, and it facilitates the water reabsorption from later parts of the tubule, thereby preventing loss of water. We call diuresis. Loss of water said to be diuresis. Hence, this water absorption process done by ADH. Hence, we call hormone as an antidiuretic hormone. An increase in body fluid volume can switch off. Increase in body fluid volume can switch off the osmoreceptor and suppress the release of ADH, antidiuretic hormone. We don't want. When water is more, we don't want ADH. ADH can also affect the kidney function by its effects. Its constrictory effects. Okay, its constrictory effects. This causes an increase in blood pressure. Increase in blood pressure can increase the glomerular blood flow. Thereby glomerular filtration rate. A fall in glomerular blood flow or glomerular blood pressure or glomerular filtration rate can activate the JG cell to secrete. Renin. A powerful vasoconstrictor increases the glomerular blood pressure and thereby GFR, and it also activates the adrenal cortex to release. Aldosterone. What is that? Angiotensin 2. It causes reabsorption of sodium and water from the distal parts of the tubule. This also leads to an increase in blood pressure and GFR, that is aldosterone. Formation of angiotensin 2 stimulate the secretion of aldosterone is a complex mechanism, and this complex mechanism is called ROS mechanism, renin angiotensin aldosterone mechanism. We can say ROS mechanism, renin angiotensin aldosterone mechanism, we can say. Then physical conditions of the body which leads to ROS mechanism. Fall in glomerular blood flow or fall in glomerular blood pressure. A fall in glomerular blood flow or decrease in glomerular blood pressure occurs due to more loss of water from the body. With more loss from the what body, these things may happen. More loss of water from body. When the blood flow is increased, factor which acts as a counter check for ROS mechanism is atrial natriuretic factor. Counter for ROS mechanism is ANF, atrial natriuretic factor, which is released from heart. Then, factor required to regulate the rise in glomerular blood flow or increase in the flow of blood is atrial natriuretic factor. Atrial natriuretic factor. It can cause vasodilation and thereby decrease in the blood pressure by relaxing vascular smooth muscles and inhibiting ROS mechanism. That is atrial natriuretic factor. ANF, which is released from the heart, right atrium of the heart. Urine formed by the nephron and ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system. Where it is stored? It is stored in urinary bladder. Carried to the urinary bladder. Here we did a mistake. No matter. Voluntary signal is initiated by the stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptor on the walls of the bladder send signals to the central nervous system. The CNS passes on motor messages to initiate the contraction of smooth muscles, and of the bladder, and simultaneous relaxation of the urethral sphincter. The process of release of urine is called, and the neural mechanism causing it called as mixturation. The process called mixturation, and mixturation reflex. Mixturation reflex. An adult human excretes an average 1 to 1.5 liters of urine every day. Urine is formed a light yellow colored watery fluid which is slightly acidic, which pH is 6.0 and has a characteristic odor. On an average, how many grams of urea is excreted out from the day? 25 to 30 grams. 25 to 30 grams every day. Various conditions can affect the characteristics of urine. Then, urine analysis of urine helps in clinical diagnosis of many metabolic disorders, as well as malfunctioning of kidney. Presence of glucose in urine is called glycosuria. Presence of ketone bodies in urine is called ketonuria. Presence of glucose and ketone bodies in urine is an indicative for this disorder. Diabetes mellitus. Other than kidney, other organs help in the elimination of excretory wastes. Other than kidney, other organs help in the elimination of excretory wastes. Lungs, liver, and skin. Then our lungs remove large amount of carbon dioxide, approximately 200 ml per minute, and also significant quantities of the water every day. The largest gland, liver is largest gland in our body, secretes bile containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with along with larger digestive wastes, along with digestive wastes in the form of fecal matter. The dash and dash glands in the skin, sweat glands, and sebaceous glands. Sweat glands and sebaceous glands in the skin can eliminate the sudden substances through their secretions, excretory substances. Sweat produced by sweat glands is a watery fluid containing sodium chloride. Sodium chloride it contains, and small amounts of urea also they have, and lactic acid even they have. Small amounts of urea and lactic acid. Primary function of Primary function of sweat glands is to facilitate a cooling effect on the body surface. It also helps in the removal of some of the wastes. Dash gland eliminates certain substances like sterols and hydrocarbon, that is sebaceous glands, and through sebaceous glands through sebum, their secretion called sebum. Secretions of these glands provide a protective oily covering for the skin, that is sebaceous glands. Wastes in small amounts which are eliminated through saliva are nitrogenous wastes. Saliva even release there are some nitrogenous wastes. Identify the given sequence for nutrient wastes. Renal artery, afferent arteriole, glomerulus, absorbed by the renal tubule, cortical fluid and medullary fluid, peritubular net, renal venule, renal vein, postcaval vein, heart. This way, nutrients will go. 218. Identify the given sequence for nutrients or nitrogenous wastes. We have to answer them as renal artery, afferent arteriole, glomerulus, Bowman's capsule, renal tubule, ureter, urinary bladder. In this way, nitrogenous wastes will go. High threshold substances will go. Which we don't want, we eliminate through urinary bladder. Which are coming back to the heart, we want. We call them as high threshold substances. Malfunctioning of kidneys lead to the accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to the kidney failure. Inflammation of glomeruli of kidney is called glomerulonephritis. Stone or insoluble mass of crystallized salts, oxalates formed within the kidney is called renal caliculi. Renal caliculi. A machine that is used to filter the blood of a person whose kidneys are damaged is called dialyzing engine. Dialyzing machine. Machine they are asking, dialyzing machine. In hemodialysis, blood of patient is taken out through and mixed with main artery. And in hemodialysis, blood of patient is taken out through main artery and mixed with anticoagulant material called heparin. Blood taken out for the dialysis is pumped into the apparatus called dialyzer. We say blood flows through the channels or tubes of this machine during hemodialysis. Dialyzer. Process of using dialysis to filter the blood of a person whose kidneys are damaged is called hemodialysis. Tubes of dialyzer are made with cellophane membrane, a transparent membrane, cellophane. Cellophane membrane of dialyzer is impermeable to macromolecules like plasma protein. The cellophane membrane of dialyzer is permeable to small small solutes such as urea, uric acid, creatinine, uric acid, and creatinine, and mineral ions. The membrane of dialyzer separates the blood flowing inside the tube and the dialyzing fluid, that is cellophane membrane. Composition of dialyzing fluid equal to the composition of blood except these substances, nitrogenous waste. Flow of material from plasma of blood into the dialyzing fluid is based on concentration gradients. If material is traveled from blood to the dialyzing fluid, it means the concentration gradient of such material A is high and B low. So high in blood and low in dialyzing fluid. Fresh dialyzing fluid should not be with any nitrogenous waste. Fresh dialyzing fluid should not be used with any nitrogenous waste. Why? If it is with such wastage, it cannot take from blood. As nitrogenous wastes are absent in the dialyzing fluid, these substances from the blood freely move out, thereby clearing the blood of its wastes. This process is called dialysis. After the dialysis, the cleared blood is pumped back to body through this vessel after adding this material. After adding pumped to this vessel vein, after adding antiheparin. Time taken for each dialysis session 2 to 6 hours. Dialysate mean dialyzing fluid. Dialyzing solution without nitrogenous wastage is called fresh dialyzing solution. We can say fresh dialyzing solution. Then dialyzing solution with nitrogenous wastage, use a dialyzing solution. Ultimate solution for acute renal failure or kidney failure, kidney transplantation. During kidney transplantation, why should we prefer functioning kidney from a donor who is a close relative? Why should we prefer? To minimize its chances of rejection by the immune system of the patient. Immune system may reject this, so that's why this method is boon for thousands of uremic kidney failure patients all over the world. That is hemodialysis. That's it. This is complete excretory products in their elimination, exclusively followed NCERT. Along with the NCERT sentences, we just followed some applications and the different dimensions of the topic also. So, practice the questions. Thank you.